With regard to UVBI, UVBI again has been used since I think 1919. UV and where it really got some of the scientific validity was with Dr. George Miley back in late 30s and early 40s. He was a surgeon at Hahnemann Hospital in Philadelphia, and in those days, this was pre-antibiotics. He was a surgeon, an obstetrical surgeon. And so he did a study, and he looked, and he was using UBI. UBI again is the blood is taken out, and it's exposed to ultraviolet light, right? And he used the full spectrum, but you can now we're using either A or C, UVA, UVC. But you can use the full spectrum. There's still some of the original machines around called NOT machines, very few. But anyway, when you do this to the blood, if you understand the blood and you look at the actual molecular structure of blood, is a red blood cell is almost identical to chlorophyll, the thing that makes plants green and that is involved in photosynthesis. And photosynthesis, as you may recall, is the sunlight hitting the plant and turning that sunlight, turning it into energy, converting it into glucose. Okay, so in order to do that, the chlorophyll must have a molecular structure that is what we call photosensitive. That is, it's able to pick up and hold onto light and to use it. And that's because of the structure of the molecule there. Well, our hemoglobin has the same thing. So it picks up the light. It's called a photoreceptive, photosensitive molecule. Picks up that light, and that light energy is used. And we found out that it stimulates, one of the things it stimulates is the endogenous or inside production of hydrogen peroxide and other things. So this UBI actually, again, stimulates oxygen production, oxygen consumption, and it stimulates the immune system powerfully. So when George Miley did his studies, and by the way, his studies were published in the American Journal of Surgery, I think in 41 or 42 after 5 or 6 years of clinical studies. And that's a regular peer-reviewed journal. But, I mean, I forget the exact details, but people that were in mild sepsis, that is bacteria in the blood after having surgery, 100% got better with 1 or 2 treatments. Those who were in moderate, which meant that they were probably, their white counts were probably, had gone sky high. Now they're probably going down because they're probably getting fatigued and they were really getting sick. Again, had 100%. And then the people that in those days they called it moribund, but it's probably what we call septic shock now, was only about 48% or something like that, which is no better than we do now, because septic shock, you're no longer dying from the bacteria in the blood, you're dying from the endotoxins that have been produced by the bacteria. So now we have different therapies for that. We have things that bind endotoxin. But again, in terms of its effectiveness at killing the bacteria, viruses, etc., it gets rid of those. It annihilates them. But it also stimulates the immune system, increases oxygen utilization, and it's a very powerful tool.